where all the complexes join, and finally uh, protein synthesis takes place. Now, ribosomes in prokaryotes and eukaryotes are different. So in prokaryotes, again, it's a ribonucleoprotein particles, but the mass of this ribonucleoprotein particles or ribosomes is 2700 kilodaltons. D is for daltons. They have a diameter of 200 angstrom units, and they have a sedimentation coefficient. Sedimentation coefficient of 70s. Now, what is this sedimentation coefficient? Sedimentation coefficient is defined by cesium chloride density gradient centrifugation. Centrifugation. It is a kind of centrifugation where in the tube, centrifugation tube, you take cesium chloride, and then at the top of it you add the molecules to be separated. So supposing if you put ribosome to be separated here, then the separation here, cesium chloride, will be based on two things. Number one, molecular mass, and number two, density. So 70S is the sedimentation coefficient in cesium chloride density gradient centrifugation for prokaryotic ribosomes. Now, the 70S ribosome can be dissociated into two subunits, the larger and the smaller. So, larger subunit is 50S and the smaller subunit is 30S sedimentation coefficient. So each of the subunit has its own ribosomal RNA and associated proteins, for example, and they can be dissociated into, their respective ribosomal RNA and proteins. So subunits are made up of both the subunits. They contain their own ribosomal RNA and specific associated proteins. Actually, more than two-third mass of ribosome is represented by ribosomal RNA. 1/3 are proteins. Okay, so if we take prokaryotic 30S subunit of ribosome, it can be dissociated into 21. It contains 21 different proteins, which are labeled as S1. S means small subunit, S1 to S21. Along with this, a single ribosomal RNA molecule, which has 16S ribosomal RNA molecule, is present. The size of this ribosomal RNA molecule is 1500 base pairs. Likewise, the larger subunit of prokaryotic 50S, can also be dissociated into its constituent RNAs and proteins. Now, there are different reports. Some books say they contain 34 different proteins. Other books say they contain 31 different proteins. So either 31 or 34 different proteins, which are then labeled as large, L1 to L31 or L34. And in addition to this, 2 RNA molecules. Instead of 1 now, in the large subunit, you have 2 RNA molecules, a 23S ribosomal RNA molecule of the size 2900 base pair, and a smaller 5S ribosomal RNA molecule of the size 1 20 base pair, they take part, in the formation of, larger subunit of prokaryotes, ribosomal subunit of prokaryotes. Likewise, you have ribosomes in the eukaryotes also. So eukaryotic ribosomes. The sedimentation coefficient of eukaryotic ribosomes is ATS. Once again, ATS can be differentiated into two subunits, 60S and 40S. The larger subunit of eukaryotes is 60S, the smaller is smaller and larger. Okay. The larger subunit is composed of, once again, ribosomes are composed of ribosomal RNA and proteins only, whether prokaryotes or eukaryotes. It's only the type of RNAs they change. So 60S subunit contains 3 ribosomal RNA, 5S ribosomal RNA, same, which was present in prokaryotes also, 120 base pair. Then you have a 28S ribosomal RNA, which is as long as 4,700 base pairs, and you have a 5.8S ribosomal RNA, which is of 160 base pairs. So these are the 3 ribosomal RNAs, but along with this, 49 different proteins, they participate in the formation of larger subunit of eukaryotic ribosome. So you can imagine how huge ribosome is, how big the molecular mass is going to be. All right, so 49 proteins, and these proteins are labeled as EL1 to EL49. E, the prefix is for eukaryotes to differentiate it from prokaryotes. In prokaryotes, larger subunits, we use just L. So here we are putting E to specify that these proteins are from eukaryotic large subunit of ribosomes. समझ में आ रहा है?
Yes, ma'am.
Yes, ma'am. All right. So likewise, you have a smaller subunit, eukaryotic subunit of eukaryotic ribosomes. And here it contains, once again, ribosomal RNA and different proteins. Only a single ribosomal RNA, which is 18S ribosomal RNA of the size 1900 base pair is present in this subunit, and there are 33 proteins. And they are labeled as ES1 to ES33. Fine. So this is the composition of ribosome in prokaryotes and eukaryotes. Now, structurally, whether it is a prokaryotic ribosome or eukaryotic ribosome, the structure remains the same. And structurally, each ribosome has 3 sites. Like if this is the ribosome, the smaller subunit, and the larger subunit, then each ribosome has 3 sites. And these sites are known as, from left, E, P in the center, A on the right. E site is known as, okay, let me start with A. It will be easy. A site is known as amino acid acceptor site. The incoming amino acid, carried on a charged tRNA will be received here. So A site receives amino acylated tRNA. or it receives amino acetylated tRNA or charged tRNA, which is carrying an active amino acid. P site is known as the polypeptide site. P site is the polypeptide site. Once, when a second amino acid is received, then the two amino acids now present are linked by a peptide bond. And this let's say two amino acid long polypeptide is then shifted to P site so as to make A site free for the next incoming amino acid. So the polypeptide would always be present at the P site. Now what is E site? E site is empty site. The polypeptide here, I'll show you all these things. We'll do the mechanism in detail. Polypeptide is always carried on a tRNA. tRNA which is specific to the last amino acid. Last or the first amino acid towards the base of the ribosome, whatever you say. If you take the 3 end, rather to say the 3 end. Chalo bataungi, dikhaungi ye. So it is always carried on a tRNA, but finally this tRNA also one by one they have to be removed, and only the tRNAs after the polypeptide has been shifted, this tRNA which does not contain any amino acid now or any polypeptide now is known as deacylated tRNA. I don't know अब इसका कर्सर कहाँ चला गया. Okay, यहाँ है. Okay. So deacetylated tRNAs. That means the tRNAs without any amino acid or polypeptide. They are shifted here. And from E site, such tRNAs then again go to the cell pool so that they are available to bind to activated amino acids again and bring them again whenever they are required to the site of protein synthesis. Is it clear, students? हो जाएगा. अगर नहीं भी clear हुआ तो ये mechanism सारा यही तो करना है. The whole mechanism is to be studied. Okay. One thing I can also mention here, I have used 30S, 50S, the S. S is known as Swedberg unit. And it is named after the scientist who discovered it, Theodore Swedberg. And a higher S value shows faster sedimentation velocity and larger molecules. Faster sedimentation velocity and larger molecules. Now, how a molecule can be large and having a faster sedimentation? It is so because the sedimentation velocity is determined by both shape and size of the molecule. And it is not actually not an exact measure of the mass. That's the reason why 70S is then differentiated into 50S and 30S, and it is not totally additive. All right. So this is about ribosomes. Then, during each cycle of translation, I don't know if you know this or not, ribosomes, okay, let me tell you this first. Ribosomes always occur in the cell in the form of subunits. They are never present as ribosome. The association of the subunits to form ribosomes, it occurs on the mRNA. When there is work to be done, they'll come together and they'll associate. When there is an mRNA in the cell to be translated, then there is a function role for ribosomes. So there on the mRNA, it will get associated. Otherwise, they will occur in the cell in the form of subunits only. So what does this mean? Now, once an mRNA has been translated, like, okay, there was an mRNA which needed to be translated. So ribosomal subunits came, joined the mRNA, formed a complete ribosome, associated ribosome. But now the time is that mRNA has been translated. So now what should happen? The ribosomes should dissociate once again because work is done now. They should go back to the cell pool. Yeah, that is what happens. Once an mRNA gets translated, the formed ribosomes, the ribosomes that were formed on it, they dissociate into subunits. Ribosomes dissociate into their respective subunits. Right? And this formation and dissociation forms one ribosome cycle. This formation of the ribosome on the mRNA, completion of the translation and their dissociation, again, this completes one ribosome cycle. And ribosomes are formed by self-assembly. You don't need any other machinery. Automatically, there are two subunits, they will self-assemble. 1968, Masayasu Nomura, a Japanese, 1968, Masayasu Nomura, a Japanese scientist. He was the first one to synthesize a fully functional 30S ribosomal subunits by mixing. Artificially he synthesized by mixing the ribosomal, specific ribosomal RNA and the proteins of it. And several years later, 50S was also synthesized in vitro conditions. In vitro means lab conditions. ठीक है जी? So this is all about the ribosomes. So we have talked about all the requirements of protein synthesis, the ribosomes, the mRNA, the tRNA, the amino acids. Now let's come to the actual process now. थोड़ा सा कुछ और भी जनरल डिस्कस कर लेती हूँ, फिर उसके बाद हम we will come to the actual mechanism of the process. Now, protein synthesis is achieved in 3 steps. Initiation, protein synthesis or translation is one and the same thing. If I say translation is achieved in 3 steps, it's okay. Initiation, so it is initiation, it is elongation and termination. Every step is important. जितना important initiation है, उतना important is to terminate at the right position. Not adding one amino acid extra. It has to be very correct. Whatever is to be kind of translated, only that should be translated. Now another thing, proteins are synthesized from amino to carboxyl end. Polar hota hai na, polypeptides are polar, there is an amino end and there is a carboxyl end. So I drew the structure of amino acid. There was an amino end and a carboxyl end. So the protein synthesis goes from amino to carboxyl end. Like you will study that DNA replication takes place, that is also unidirectional. This is also unidirectional. DNA replication takes place in 5 to 3 direction. Protein synthesis takes place from amino to carboxyl end. So it is unidirectional. Always remember, these are the concepts, you know. Actually, undergrad is for developing the concepts, and then postgrad and later on you apply these concepts and you study the applications and other things. So if your concepts are somehow weak and you mix up things, then it will be very difficult to handle things later. And that's how you should be very sure about developing clean concepts, and all these things will be just taught to you in your once, you know, this time in your lifetime. Nobody is going to teach you these concepts again, such basic and such, you know, in a such simple way and starting from scratch. Nobody is going to do it again. So, and I told you in the first class only, and you will have a feel also that this paper is tough. It's not an easy paper. So you need to be regular, attentive, and try and understand and studying yourself. That is going to help you, because teachers will do their part. They'll try to simplify things, but then retention is your hard work. How much you retain, how much you remember. Anyways. Okay. ठंडा ज़्यादा है. It's though अभी तो class थोड़ा late है सुबह, तो it was really cold. Anyways, where is the cursor? ये this I think device में कुछ गड़बड़ हो रही है मेरी. I am unable to find the cursor. Okay. या मेरी spectacles, they need to be changed. Okay. So protein synthesis is unidirectional. Proteins are synthesized from amino to carboxyl end always. And for that matter, mRNA is translated from 5 to 3 end. The polarity is maintained there also. All right. Then there is another important feature, that translation can begin, it can start while. Dekho, this is my original writing, but how dirty it comes on this, because the you know, this digital device is so slippery that it's hard to kind of hold your hand. Translation, sometimes, you know, I'm able to, so that natural writing comes up. Otherwise, when I look at my write-ups, they look so clumsy. So translation can start while the mRNA is still translating. Still transcribing. I'm sorry. बातें करने में. Transcribing. When mRNA is being produced, transcription is taking place. What I want to say is, let's say this much part of mRNA, 5 end of mRNA has been synthesized. The rest of it is yet not synthesized. It will get synthesized. And I don't know. आपको मालूम है mRNA synthesis कैसे होता है? I don't know. पता होगा कि नहीं. This is how. This is a double-stranded DNA. It will open up. You have a transcription bubble. If you have studied it or not, I don't know. So this is the transcription bubble. So this is the transcription bubble where the DNA has opened up. So here, you know, then RNA polymerase will come and bind, and it will start synthesizing mRNA. This bubble will keep on moving. This much part is synthesized now, mRNA is synthesized, then it will move ahead and rest part of mRNA will be synthesized. So this is the 5 end of the mRNA. What I want to say here is, mRNA synthesis is still taking place, but whatever the part has been extended out, the ribosomes can come and bind to it and they can start translating it. So mRNA and mRNA synthesis, that means transcription and translation, can be coupled. They can, I told you in the beginning only. So this is coupled transcription and translation happens in prokaryotes. So in prokaryotes, the ribosomes can join to mRNA that is still getting transcribed. I think this is the best way to explain this. So to the 5' end of the nascent, this is the term, nascent, 5' of the nascent mRNA. Nascent mRNA means mRNA that is getting newly synthesized. The ribosomes can come and join and they can start translating it. So as to show that in prokaryotes, transcription and translation are coupled. This does not happen in eukaryotes. Now, further, in prokaryotes, what another interesting aspect is, many ribosomes, many ribosomes can simultaneously at one time join a single mRNA. Look, I also made two. So many ribosomes will simultaneously join a single mRNA molecule, and they will kind of go on translating it. This happens to increase the efficiency of translation or mRNA utilization. Efficiency of translation or mRNA utilization. Understand? So the group of ribosomes that associate with a single mRNA is known as polyribosomes. So the group of ribosomes that are associated with a single mRNA molecule are known as polyribosomes. Now, interesting thing is each ribosome will be translating independent of the other, works independent of the other, and gives rise to its own polypeptide. So 5 polypeptides, if 5 ribosomes are attached to a single molecule, some starting from, some present at some length of mRNA, others present at others. So because ribosomes are macromolecules, ribosomes are huge. They are macromolecules. So one ribosome per 80 nucleotides. That is the density that is allowed, maximum density, because ribosome takes space on the mRNA. So only after 80 nucleotide pairs of mRNA, another ribosome can sit, because that much space of nucleotides is occupied by a ribosome. Now the polysomes, polyribosomes or polysomes, they are also known as polysomes or polyribosomes. Many ribosomes that are associated with a single mRNA molecule. So polysomes synthesizing hemoglobin protein. Hemoglobin protein. Hemoglobin protein is approximately 145 amino acid and about 500 nucleotide pairs of mRNA. So typically 5 ribosomes, they join here to one mRNA molecule. So 85s are 400, so that is the density that has been seen. So these are some general features associated with mRNA. So if then It is though obvious to understand this, but then I will just make a line diagram. Supposing this is mRNA, single stranded mRNA, 5 end to 3 end, right? Now, ribosomes have joined to it. So, प्रॉब्लम तो करता है नहीं. 1, 2, 3, 4, and 5, let's say. Now, look, this is the ribosome that has joined latest. This is the oldest. ये सबसे पहले join किया था. And it has already, and direction is this. So this has already, this ribosome has already traveled this much length on the mRNA, and it is about to reach to the 3 end, right? So the polypeptide produced by this is polypeptide. This will be the longest because so much length, this much length of mRNA has been traveled by this ribosome. The polypeptide will be shorter. For this will be even shorter. This will be even shorter, and this has just begun. Understand? I hope it is understandable. Am I audible, students?
Yes.
All right.
Yes,
so with all these basic informations, then translation, we can start studying about translation. And as I told you, we will be studying it. It happens in 3 steps, and we will be studying it also in 3 steps. That is initiation, elongation, and termination. All right. So let's begin with the 20 मिनट तो हैं अभी, कुछ तो शुरू हो जाएगा, जितना भी हो जाएगा. Okay, let's start with initiation. Good thing is the class is at 11:40, so we have a break का time. 5-7 मिनट ऊपर नीचे कोई फर्क नहीं पड़ता फिर. Okay. So initiation of translation happens by formation of an translation initiation complex. So initiation. Kiski initiate? I am only teaching translation, so there is no chance of mixing up. So if I say initiation, of course, I am talking about initiation of translation. So this takes place by formation of a translation initiation complex. Now, what all are required for forming this initiation complex? Intact 70S ribosome. That means both the subunits should be there. Then an mRNA that is to be translated should be there. Then you should have an FMET, formulated methionine, carrying tRNA, and that has FMET attached. FMET, formulated methionine, the first amino acid to be transferred in prokaryotes on the P site. So we need this, and for initiation we need an empty A and E site on the ribosome. So if this condition is met, we say translation in prokaryotes is initiated. For this initiation, for all this to happen, 3 initiation factors are required. We will do the same in eukaryotes also. Abhi I am starting with prokaryotes. So all this I am talking about prokaryotic system. So 3 initiation factors are required, and they are IF1, IF2, and IF3. Initiation factor 1, 2, 3. Each one of them binds to a specific site. binds to a specific site in the ribosomal subunit. The specific sites to which the initiation factors bind, then they will become E or A site of the ribosome. So that means the initiation factors, they bind to E and A site only. Initiation factors bind to E and A site only. Okay, let's see where exactly they bind and what is their function. Now, IF3 binds to a site in ribosome which will eventually become E site. This site will become E site. So that means IF3 is specific to bind to E site. Why it binds to E site? Because its function is to prevent binding of 50 binding of IF3 prevents 50S to join 30S. The function is to prevent 50S from joining to 30S. So that means if 50S is to join 30S, IF3 should be removed. So that means the binding of IF3 is giving time for whatever has to happen before the whole ribosome is formed. Are you getting my point?
Yes, ma'am.
Otherwise, when I'll do it, things will become more clear. As of now, it just prevents 50S to join 30S or formation of a complete ribosome. That is the function. IF1. IF1 binds to a site in ribosome that eventually becomes A site. So it is specific to bind to A site, that means. All right. And once occupied the site, once occupied by IF1, then this site is not free for initiator tRNA. That is FMET, tRNA, FMET. FMET is formulated methionine. I don't know, I can't see the cursor. मेरी या तो आंखों को कुछ हो रहा है. This is formulated methionine and this is the tRNA which is specific to carry this methionine. So they are complex. That's how a dot is put in between FMET dot tRNA FMET. In other words, it means that this is a tRNA which is specific to carry FMET and this is carrying FMET now. FMET is formulated methionine. So this is that, activated, charged tRNA, adenylated tRNA, right? Okay. So IF1, the site that is occupied by IF1 will become the A site. Once A site is occupied by IF1, this site is not free for initiator tRNA. So it prevents of binding of initiator tRNA to the A site. Okay. Then IF2. IF2 binds over IF1 at the A site. And it is basically a GTPase protein. It is a GTPase protein. Do you understand what is GTPase? Any protein that will break GTP into GDP and PI, and this will release energy. जैसे ATP is broken down into ADP and PI and energy is released. So GTP is also broken down into GDP and PI, inorganic phosphate, and energy is released. and it is a GTPase proteins which hydrolyzes. This is breaking down or hydrolysis of breaking down or hydrolysis of GTP. Okay, then it binds over IF1 at the A site. So it binds over IF1 at the A site, and this factor IF2 is able to interact with all the three. That means the small subunit, which is 30S for prokaryotes, IF1 because it is binding over it, and charged initiator tRNA. Charged, it is able to interact with all three. Initiator tRNA, that is FMET, FMET dot tRNA, FMET. So at the three sites on the ribosome, E, P, and A, E gets occupied by IF3, P is empty, and IF1 is present on the A site, over which IF2 is attached. So this is the sequence of events that happens. Now the same thing I will show you with the help of a diagram, and things will become all clear. इतना ही हो पाएगा, बस diagram बनेगा. Okay, कोई बात नहीं, बनाए. So we are starting from a previous cycle where the ribosome is present on the mRNA and translation is taking place. So you have A site. ऐसा बनाएंगे, मैं पहली बार आपके सामने बना रही हूँ, आप भी ऐसे बनाएं, फिर आपको आदत पड़ जाएगी. This is E site. So E, P, and A. Okay, let's say this is 70S ribosome. So after completing one ribosome cycle, this will dissociate into a smaller subunit, which is 30S. So this is 30S, but it will have the three subunits as we just defined, EPA. So E, P, and A, and it will, the other part will be the larger subunit, which is 50S. Once again, it will have E, P, and A. E, P, and A. So this is 50S. All right. So now, first of all, what all the initiation factors are binding to 30S. That's what we have done. Now, let's begin with 30S. So this is our 30S now. The E site, the P site, and the A site. Now, in the first step, what happens is IF3, which can be represented as a circle with 3 written in between, because it will be easy for us to place. So this comes and this binds to the E site. So this is E, P, and A. So this is number 3. So this attachment takes place. Now, I told you the role of IF3. IF3, if it is bound to 30S, then 50S cannot come and bind to it. Yehi role tha na?
Yes, ma'am. Yes.
So look at this. This 50S now cannot bind, because अगर ये अब भी bind कर देगी तो बाकी चीजें तो अभी कुछ हुआ ही नहीं है, ना mRNA bind हुआ ना कुछ हुआ, right? So it cannot bind as of now. So there is an obstacle in the form of IF3. Otherwise 50S can come and bind to it. So it is preventing the binding of 50S, right? Okay. Now to this complex now, then IF1 comes and binds. Okay. I'll take it forward. Okay. So IF1 is specific to bind to A site. Fine. So the same thing we'll make again. E site, P site, and A site. E, P, A. So to the E site, 3 is already bound, and IF1 is specific to bind to, sorry, this is A, P दिख रहा है ये भी. This is A site. So 1 binds to A site. Now what is the function? Once IF1 is bound to the A site, then IF2 will join over it. IF2 is always bound to a GTP molecule. So this will come and bind over IF1. In the next step, I'll show you. This is positioned in a way that it reaches to the P site and is also able to interact with FMET tRNA FMET that will join the P site. You know, the first FMET is delivered on the P site. All the subsequent amino acids are delivered on the A site. I hope we know this. Bachcho?
Yes, sir. Yes.
The first initiator tRNA carrying the initiating amino acid, which is now we are leaving the P site empty. So to this now IF2, which is actually IF2 joined with a GTP. So IF2 and GTP, they join. And this happens over the IF1. So you have E site, you have P site, and you have A site. E, P, A. 3 already joined, 1 joined here, and now 2 linked to a GTP molecule joins over 1. Now look at this. इसके बाद अभी 50S नहीं ज्वाइन करेगा, अभी mRNA सेल के पास चॉइस है. Now we have this as the complex. This is the complex. यहाँ से आगे बढ़ते हैं. Now सेल के पास प्रोकैरियोट्स में there is a choice. Either mRNA can join now. So mRNA doesn't come and join to the complete ribosome. You will see this joins to the 30S subunit. फिर उसके बाद जब सब हो जाएगा, ये सब हटेंगे, फिर 50S आएगा. Either mRNA or the tRNA carrying FMET, tRNA, FMET. This can join. कोई भी पहले join कर सकता है, दोनों को ही join करना है. So anyways, this is mRNA, 5 single-stranded RNA molecule, and your tRNA. Oh, मैंने tRNA नहीं कराया ना आपको structure. tRNA का structure भी बता देना चाहिए था. Anyways, this is L-shaped structure. This is, here is present FMET. So this is what? FMET, tRNA, FMET. कोई कराएगा दूसरे पार्ट में, don't worry. ऊपर के पार्ट में RNA DNA का स्ट्रक्चर होगा ना, तो tRNA का स्ट्रक्चर वहां पे कराएंगे. क्लवर लीफ मॉडल स्ट्रक्चर, एंड देन this is 3-dimensional structure. Clover leaf model is a 2-dimensional structure. This is 3D structure of tRNA, which is an L-shaped molecule. So this is FMET, tRNA, FMET. ठीक है, दोनों में से कोई भी ज्वाइन कर सकता है. In any case, दोनों को ही ज्वाइन करना है ultimately, तो right? So the complex that will be formed now will be the initiation complex. अब यहाँ पे initiate हो सकता है ट्रांसलेशन हमारा. Anyways, आराम से बनाते हैं. E, P. I hope you are using your hands and your notebooks.
Yes.
Yeah.
Yes, ma'am.
3. So you have your 1 and you have your 2, and on the P side then your tRNA. This is your tRNA. This is your tRNA carrying FMET, and this is the GTP. So look at this. 2 can interact with 1, it can interact with the tRNA also. Right? So here you have the anticodon. Then mRNA also joins. This is mRNA. This is mRNA joins. 5 end of mRNA, 3 end of mRNA. So anticodon, this is the anticodon of tRNA. This fuses with the codon, first initiator codon, AUG, let's say. Let's say this is AUG of mRNA. They base pair so that this tRNA then by hydrogen bonding, it binds to the first codon. So base pairing takes place between codon, initiator codon, AUG of mRNA and the anticodon of FMET, tRNA, FMET. Methionine is the first amino acid to be transferred, right? And it is formulated in prokaryotes. In eukaryotes, it is not. That is how it is known as FMET, formulated methionine. So this complex is known as the initiation. No, this is not known as the initiation complex because 50S has not yet joined. Now, look at this. What will happen? I'll take 5 minutes. I'm sorry, ये हमेशा चलेगा तुम्हारा. इस दिन तुम्हारी क्लास ऐसे ही जाएगी, आपका ब्रेक जाए, मेरा भी जाता है, मेरी भी इसके इमीडिएटली बाद क्लास होती है. Anyways, let's not waste time on that. IF3 का फंक्शन यही था. IF3 was required so that this could associate. जैसे दो साइड से प्रोटेक्शन चाहिए, you can function is over. The first tRNA has joined the ribosomal 30S subunit. Now IF3 can leave because bonding between codon and anticodon has already taken up. अब इसका कुछ नहीं, टूट नहीं सकता ये. It will stay there. So IF3 leaves. The leaving of IF3 allows. आप arrows देख रहे हैं? This is coming in the reaction. 50S joins. 50S with E and P and A. This joins now, right? And with this joining and binding of 50S, then the initiator complex is completed. So this is 50S. Here is 30S, and then you have E, P, and A. So you have your A, P, sorry, अब इसको क्या हो गया फिर से? Okay. You have E site. Here you have the FMET tRNA. FMET tRNA. In between is your mRNA. 5 end to 3 end. They are base paired. 1, 2, 3. 3 hydrogen bonds. 2, 3. Because codon is triplet of 2 and 3. Okay. And here you have on A side you have number 1 attached, and to this you have number 2 attached with a GTP molecule. Okay, now 50S has joined. इसके बाद क्या होता है? GTP hydrolysis takes place. GTP is hydrolyzed to GDP plus PI. इसके हाइड्रोलिसिस होते ही IF2 निकल जाता है बाहर, IF2 leaves. IF2 leaves, उसके बाद IF1 also leaves. अब इनका काम खत्म. You know, the point was they were required for assisting addition of the first tRNA. So that is already bonded now. So their function is over. So now the ribosome, complete ribosome has formed, and it looks something like this now. Initiation has taken place now. Rather, इसको बड़ा बना देते हैं because this is empty now. And on the P site you have your tRNA, and the A site is also empty. This is A site. And here you have the tRNA carrying FMET. No initiation factors now, and you have a mRNA. 5 end to 3 end. So this is FMET. So this is known as 70S initiation complex. This is known as 70S translation initiation complex. So what does it have? It has an empty E site, an empty A site, कोई factors नहीं, no factors. This is empty, so that next incoming amino acid can come here. And when this comes, then this is elongation. Then it is not initiation. Initiation has been achieved by transfer of FMET on the P site. Now, first amino acid, that is formulated methionine, is always transferred on the P site. Remaining all amino acids will be transferred on the A site, because then now on you will have a polypeptide lying on the P site. That's how it is known as the P site. Clear हुआ बच्चों? Clear हुआ?
Yes.
Okay, just 1 second and 1 second. Before the joining of 50S, this complex, ये वाला complex is known as 30S initiation complex. This is known as 30S initiation, translation initiation complex. Before 50S joined. This complex is 30S translation initiation complex in prokaryotes, because mRNA is joined, FMET is also joined. So we are able to form an initiation complex in prokaryotes. ठीक है बच्चों? हाँ जी.
Yes, mommy.
Okay. कुछ और चीजें I'll talk about it. थोड़ा थोड़ा और भी कुछ मैं इसके बारे में बताऊंगी अब अगली क्लास में. अभी I'll take attendance. एक घंटे में ना थोड़ा पेपर लंबा है, कॉन्सेप्ट्स लंबे हैं, बट